Hello and welcome to Nikolai's genetics lessons, and this video going to be about breeding and selection. And in this video I'm going to explain how by breeding two pure lines of animals, it could be also plants, it doesn't matter, we can find how many genes control the trait. So here's a problem. In rabbit variety 1, ear length average 4 inches. In second variety it is 2 inches. Hybrids between these two varieties average 3 inches in ear length. When these hybrids are crossed to each other, the offspring exhibit a much greater variation in ear length, ranging from 2 to 4 inches. Of 500 F2 animals, 2 have ears about 4 inches long and 2 have ears about 2 inches long. Approximately how many allelic pairs are involved in determining ear length, and how much does each effective gene seem to contribute to the length of the ear? What do the distributions of parental first generation and second generation probably look like? If you think that you can solve this problem, I recommend you to pause video here, try to solve this problem on your own first, and when you would be ready you can run video again and you can compare your answers with my answers and explanations. So in order to solve this problem, everything you have to know is a formula. Of course you can solve this problem without a formula. I will show you later how to do it, but first I want to give you a simple solution when you can find pretty fast an answer using a formula. And here is a formula. This formula shows occurrence of the extreme genotype, and 1/2 raised n and raised 2. This is a distribution of the extreme phenotype. So let's check our problem. We told that we have in F2 generation animals with two extreme genotypes, one with ears that is 8 inches long and another with 2 inches long. We have two animals out of 500. Two have ears about 4 inches long and two have ears about 2 inches long. So occurrence, as you see, 2 out of 500, or we can simplify as 1 out of 250. So now we have left part of the formula and right part of the formula. So what we can do in the next step, we can divide 1 by 250, and this is how we would find decimal number. And decimal number would be 0.004 equals to 1/2 raised n and squared. Now let's get rid of this square here. So what we have to do, we have to take a square root from both sides. And what we are going to get here, on the left we are going to get 0.063. That would equal to 1/2 raised n. Now we have to solve for n, and n here in this formula stands for the number of allelic pairs that control the trait. So how we are going to do it? We have to find reciprocal of each number. What is reciprocal? Reciprocal has every number, and this is a number. When we multiply this number by reciprocal, we are going to get 1. So for example, reciprocal of 5 would be 1/5 or 0.2. If we multiply 5 by 0.2, we are going to get 1. Or reciprocal of 0.5 would be 2. if we multiply 0.5 by 2, we are going to get 1. And reciprocal of 2 would be 0.5. So as you see, this is very easy. So in order to find reciprocal of this number, we have to divide 1 by this number. And then we would find the reciprocal of this number. And reciprocal of 0.063 would be 15.87. And reciprocal of 1/2 would be 2. So 2 raised n equals to 15.87. So now we can easily find n. So how many times we have to multiply 2 by itself in order to get this number, or this number is rounded and can be even rounded to 16. So as you see, we have to multiply 2 by 2. We are going to get 4. If we multiply by 2, we are going to get 8. And if we multiply by 2, we are going to get 16. And this is very close to this number. So we have found number n that is equal to 1, 2, 3, 4. So we have to multiply 2 by itself 4 times, or 2 raised 4 would equal to 16. So this trait under the control of 4 allelic pairs, or 4 genes. And I want to tell you that this rule only applies to such traits that is quantitative. Traits that is qualitative, we cannot find number of alleles that control the trait using this formula, because quantitative trait is such trait that is under control of many genes, and each gene and each allele would contribute something to the trait. So for example, quantitative trait would be for example, your body weight, or your tallness, color of your skin, or color of your eyes or hair. So as you see, qualitative trait is something that can be in two states. For example, with for example, rabbit can be black or white, or plant flower can be red or white, for example. Another example from the plants kingdom. And of course, our problem is quantitative problem, because length of the ears would be a quantitative trait, because many genes would contribute to this trait, and we just have found that 4 genes or 4 allelic pairs contribute to this trait. And now we can answer other questions, and we can list genotypes of the parents and genotypes of the all the following generations, F1 and F2. So because parents has 2 extreme phenotypes, we can suggest that parent 1, whose ears is 2 inches, represent one extreme, and parent 2, whose ears 4 inches, represent other extreme. So we can say that the first parent would be small a, small a, small b, small b, small c, and small d, small d. And genotype of the second parent would be capital A, capital A, capital B, capital B, capital C, and capital D, capital D. And this explains that when we cross these 2 parents, so parent 1 and parent 2, or 100% of the F1 generation would have uniform genotype and uniform phenotype length of the ears. In this problem, we ignore environmental influence just for the ease of explanation. So our trait under the control of 4 genes: gene A, gene B, gene C, and gene D. And of course, second parent also has the same genes, but the second parent would have different alleles. So that explains why this parent has different length of ears. And for the gene A, parent 2 would contribute dominant allele A. So we can put dominant allele A. There is no other choice. He has two dominant alleles A. So parent 1 would contribute recessive allele A for this gene A. And there is no other choice for the parent 1. Both his alleles for the gene A are recessive. So that's explanation why in F1 generation for the gene A, all F1 generation going to be heterozygous, and the same rule applies to each allelic pair. So F1 generation going to be heterozygous for all 4 genes or allelic pairs, for the gene A, gene B, gene C, and gene D. And as you remember, here we have 2 inches, here we have 4 inches, and in F1 generation we have ear length 3 inches. So how we can find how much each allele contribute to the trait? This is very easy. So as you see, parent 2 has 4 allelic pairs or 8 alleles that produce phenotype with 4 inches. So that means that we have to divide 4 by 8, and this is how we find that each dominant allele contribute 0.5 inches to the ear length. So each dominant allele. Let's now find how much each recessive allele contribute to the ear length. And once again, we have 2 inches and 8 alleles that contribute to the ear length. So this time we know that each allele contribute 0.25 inches to the trait. So for example, this would be dominant allele and this would be recessive allele. So each dominant allele would contribute 0.5 and each recessive allele would contribute 0.25. And now, of course, we can find easily length of the ears in F1 generation. We just have to add 0.5 for dominant allele plus 0.25 for each recessive allele, 0.5 here, 0.25 here, 0.5 here, plus 0.25 here, 0.5, and 0.25. If we add all these numbers, we are going to get 3 inches. So what is the distribution going to be in F2 generation? I will show you this on the example of 1 and 2 allelic pairs, because to show you on the example of 4 allelic pairs, this would be too time consuming, and anyway you can have an idea even if I would show you example with 1 or 2 genes and allelic pairs. So if this trait would be under control of one allelic pair, and parent 1, for example, would be small a, and parent 2 would be capital A, and as you see, all the F1 generation going to be heterozygous. Capital A small a, capital A small a, capital A small a, and capital A small a. So this is what we see here in F1 generation. But here we had example with 4 genes, 4 allelic pairs, and we reduced our example to parent 1 with 1 allelic pair and parent 2 also with 1 allelic pair. And 100% of the F1 generation would be heterozygous, just like in this example, but in this example it is heterozygous for each gene, each of the 4 genes, and here we have 1 gene, 1 allelic pair. So what would happen if we self cross this F1 generation? We are going to get F2 generation. So parent 1 would be heterozygous. We take this parent from F1 generation. We have only one choice, heterozygous. We take parent 2 also from this generation, only one choice. Parent 2 also have to be heterozygous. So when we build a Punnett square in F2 generation, distribution would be as follows: capital A here, capital A small a here, capital A small a here, and small a here. So if in this example we see that 100% of the progeny is genetically uniform, here we have a distribution that is different from F1 generation, so this is F2 generation, and distribution is 1/4 or 1/4. We have genotype that is homozygous dominant, so 1/4. 2, we have 1/2 or 2 out of 4 that is heterozygous. We can say that 1/2, because 2 out of 4 is 1/2. And as you see, 1/4, we also have homozygous recessive genotype. So our distribution would be 1/4 to 1/2 to 1/4. But what would happen if we would have 2 allelic pairs? If this trait would be under the control of 2 genes, gene A and gene B. Once again, I'm not going to show you distribution of 4 genes because this is time consuming. This would be my second example, and you would know how to solve such problem even with 3 and 4 allelic pairs. So next example would be with 2 allelic pairs. So one parent would be once again homozygous recessive, so small a and small b, and we cross with another parent who is going to be homozygous dominant for each allelic pair, so capital B, and capital A. And if we cross once again these 2 parents, so parent 1 and parent 2, this is parental generation, we are going to get F1 generation that is going to be capital A small a, and capital B, small b genotype. And once again, if we need to get F2 generation, we have to find all the possible gametes that this genotype can produce, and the first variant of the gametes would be capital A, capital B, so capital A, capital B. The second variant would be capital A and small b, so capital A and small b, and the third variant would be small a, capital B, small a, capital B, and the last variant would be small a and small b, so small a and small b. So this would be all possible gametes, genotype of the gametes for the parent 1, and when we cross parent 1 with parent 2, and because this is F1 generation, as you remember, in F1 generation we have only one genotype for the whole F1 generation. So in the case of two genes, both genes would be in the heterozygous state. So when we cross one parent with another parent of the same genotype, that means that here we have to show genotype of the second parent, genotype of the gametes that is going to be also the same, capital A, capital B, capital A, small b, and small a, capital B, and small a, small b. Now we can build a Punnett square, and we would find distribution of the genotypes and phenotypes. So we are going to get a square 4 by 4, and genotype of the gametes of the parent 1 would be on top, and gametes of the parent 2 would be here on the side. And as you see, we cross two parents with the same genotype. We cross two parents from F1 generation. But in F2 generation, so this is F2 generation, we are going to get distribution of phenotypes. For example, here we would have one extreme genotype that is going to be capital A, capital A, capital B, capital B. Here we would have another extreme genotype that is going to be small a, and small b, small b. And of course here we would have distribution of different genotypes, capital A, capital A, capital B, small b. As you remember, dominant allele gives 0.5 to ears length, and small allele 0.25. So this genotype would produce slightly shorter ears than this genotype. So this would be extreme and extremely large. And another genotype would be small a, capital A, and capital B, capital B. And as you see, this genotype equals to this genotype, because here we have 3 dominant alleles and 1 recessive, and here we also have 3 dominant alleles and 1 recessive allele. And here we have small a, capital A, and capital B, small b genotype. This time we have 2 dominant alleles and 2 recessive alleles. So now we just complete our table, and as you see, this takes a time even with 2 genes. That's why I cannot show you the same table with 4 genes, because this table with addition of each allelic pair would be 4 times bigger, and that means with addition of two allelic pairs, our table would be 4 times bigger, then one more time 4 times bigger, or we have to multiply this 16 variants by 4, and then so we are going to get 64, and then we have to multiply once again by 4. So we are going to have 256 cells. So as you see, this is a rather time consuming to find all the distribution of the genotypes for such genotype that includes 4 or controlled by 4 allelic pairs. So here we would have capital A small a and capital B, capital A small a and capital B small b, small a, capital B, small a and capital B small b, and capital A small a and capital B small b. We're almost done. Capital A small b small b, and small a and capital B small b. And once again, this is one extreme, here is another extreme, and we can find how many different variants we have here. We just have to find, for example, here we have 3 dominant alleles and 1 recessive, and here we have also 3 dominant alleles and 1 recessive, and here we also have 3 and 1 recessive. So we can circle them with the same color. So all these different genotypes would produce the same phenotype because it has 3 dominant alleles and 1 recessive allele. One more here. And if we use once again different color, we can find those genotypes with 2 dominant alleles and 2 recessive alleles that also would produce the same phenotype. And one more here, one more here, and this one, and the last variant would be when we have 3 recessive alleles and 1 dominant allele. So this is going to be one genotype, second and third that produce the same phenotype. One more here, and I missed this genotype that produce 2 dominant alleles and 2 recessive alleles. So I have to color it in the blue color, and let's count how many different phenotypes we have here. 1, 2, 3, 4, and 5. And our distribution would be as follows. So we have 1 extreme phenotype, extreme large. So 1, 2, we have 1, 2, 3, 4. Next phenotype represented by 4. And next phenotype would be 1, 2, 3, 4, 5, 6. So 1 to 4 to 6, and next phenotype would be 1, 2, 3, 4, and this is going to be frequency. And the last one is extremely small, so the phenotype is occurrence of this phenotype is 1. So here's our distribution: 1, to 4 to 6 to 4 to 1. Now you see that this is one of the longest videos, and if I would give you example with all 4 allelic pairs, I would get a square that is going to be 16 by 16 or with 256 cells, and this would be too time consuming to show you all the distribution of the phenotypes. But now you have idea how to solve such problems, and this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.